variants of Cas9, different variants of CRISPR-Cas and its applications, limitation, advantages. You must all be familiar with this. So now we will be focusing on how to design a CRISPR-Cas experiment yourself. So all of you are in PhD, or let me, if masters. So how many of you are from PhD? Okay. So you are all working in the field of plant or? Okay, plants. Okay. Okay. So we'll try to take some examples from human as well, human genes as well. So before designing the single guide RNA, you need to select the appropriate, select first the appropriate vector, because uh, the selection of vector depends on the target organism which you are going to plan to target. Like if you are targeting a plant, then the promoter and the expression cassettes will be different. So you will take the expression cassette specific for plant, and in case of animals, you will take the promoter and the expression cassette specific to that particular organism. So this is the thing you should have to keep in mind first. And after that, the second point is delivery options. So it depends whether you want the CRISPR-Cas system to integrate stably, or you want the transient expression of it. Depending on that, you will select your vector. And if you are looking for a stable expression, then the CRISPR-Cas will integrate itself in the plant chromosome if we talk about the plant. So here are some examples of CRISPR vectors specific for plants. Similarly, we will talk more about in vectors specific to human scenario. So where can you get these specific vectors? There are various repositories which are available online where you can purchase these specific vectors based on your requirements, and one such repository is AdGene. You all can just type AdGene in Google and you can just click the first link, and then you can be able to reach here in this homepage. So this is one of the repositories where you can purchase the CRISPR specific vectors or any vector at a minimal cost. Please let me know if you have reached till the home page. Okay. So here in the search bar, you can just type CRISPR vectors or CRISPR and then search it. And with this search, it will give you some results. Like in this case, if you type CRISPR vectors, you can get a list of 4,319 available plasmids in this repository. And similarly, you can click that these in the catalog, and after that you will get to open this page, and here you can see these filter. These are various parameters which you can alter according to your needs. And if you are working on plant expression system, then you can select here plant expression, and you can see 213 vectors which are available from which you can choose. And similarly for bacterial expression, we have 401 vectors, and for insect and mammalian expression, we also have vectors. You can tweak these parameters and you can easily get the vector which you are looking for. you can just scroll through if you want to. You can easily see a list of vectors with various different specifications. Some of them are having Cas9 as endonucleases, and some are having Cas12, and you can further search it down. So based on this, you can select a particular vector which will be required for you. And here I have made a list of vectors which are specific for plant, and some of them we have used in our lab as well. So PHSC 401, PKSC, and so these are the vectors which we have used, and PRGEB 31. So these have different specifications. So this PHSC 401 has hygromycin resistance, and this PKSC has kanamycin resistance, and similarly different configurations of these vectors are available. So once you have gone through this, you can make a list of your specific vectors that you can opt for, and you can simply purchase it by logging in, and with the minimal charging they can provide you vectors. So these are different vectors, different variants of Cas and different PAM protospacer adjacent motif which are required for designing our single guide RNA. So here we will talk more about this Streptococcus pyogenes Cas9 variant with PAM sequence of NGG. Because we have worked on with this as in PHSC 401 and PKSC 401, Streptococcus pyogenes Cas9 is there, so that's why we are emphasizing more on this. And till now you have learned that how to select an appropriate vector on AdGene based on your requirements. So what's next? The next is the selection of target gene, because you want to target a particular gene that has to be there, which is going to have a particular function in either plant or animal, whatever you are targeting. So for this, the sequence for that target gene should be known, because otherwise you would have to get it sequenced, and you have to know the legit sequence for it based on homology basis. Like you can blast search and find out the particular homologs associated with that genome if the genome is available. But the main thing is that the gene sequence has to be known. And to retrieve the gene sequence, there are various databases which are available. To name a few, and the common ones are NCBI, Ensemble, Ensemble plants. I hope all of you are aware of this. So we will try to retrieve and give you a live demo of it. So all you can open NCBI homepage. So you can type in any gene you want, and those of you who are not familiar with it, you can select from this drop down to gene, and you can type any gene specific to plant or bacteria or animal, whichever you want. And here I'll be giving you example of this phytochrome interacting factor 3, which is a gene involved in the growth and development of plant. So this is an example. You can take your own sequence as well, and either you can just type this. Okay, so after typing the name of the gene, you can get the list here, and once you click the top this gene ID, then you will be forwarded to this page, and we have to take the coding sequence. Is that clear? Because exons will be targeting exons, so we'll taking the coding sequence. So after selecting, you can just scroll down, and in the reference section, you can find gene written or CDS written here. You can just simply click on the CDS. Okay. Okay. So you can type any name of any gene. So the first gene ID or the gene you get, you can just click it after selecting the gene here. Otherwise, it will open a lot of things which we don't want. So you can just click it, and once you clicked it, you will reach this page where all the description about this gene will be there, and you can just scroll down. If you scroll down, you will get to know these things written, and if you further scroll down, you will able to see the gene and the CDS written here, from where you can retrieve the sequence. So basically what I'm telling you is that this is the retrieval of a gene part. You need a sequence for designing single guide RNA. Okay. Okay. So once you have opened this NCBI database, all must have opened it. So you can select from this drop down, or I can just open simultaneously with you. Okay. So now from this drop down, instead of all databases, you have to select the gene here. Okay, selected. Then you can type PIF3, the gene which I'll be giving example, or you can take your some other gene as well. So then you can search, and then you will get a list, and click on the first gene ID which you will get, and after that you have to scroll down, and okay, I think I got what's your confusion. After scrolling down, here you will see mRNA and proteins written here, and in that NM is our mRNA and NP is our protein, so we want this, so we will click on this, and below are the isoforms, but we don't want to select that, so we'll select on the first one. So okay, then you will get here. Everybody is on the same page? Okay. So from here you have to again scroll down, and then you can get gene written here and the CDS here, and this is our sequence of this gene, phytochrome interacting factor 3. So if we click on the gene, then it will highlight the whole sequence, but we want only the CDS, so we will click on the CDS, and once we click the CDS, as you can see that it will only highlight the coding sequence. So at the top right bottom you have FASTA written here, so you can just click it, this will take you to the next page where this will having our CDS sequence for this PIF3, and you can just simply copy it and paste it in a Word file. For your reference. So as the sequence have to be in FASTA format, so we'll add the identifier first and write PIF3. We didn't copy the sequence as it is, as there was a very long header, so we have just simply written it PIF3. So once you have pasted, please let me know, then we can proceed. Okay. Then this was the sequence retrieval part. So now you have a CDS sequence for which you want to design the single guide RNA. So in the Word file. Okay. Uh, you have to provide it a header so you can write anything like with a greater than sign. Okay. You are doing with the same gene? Okay, okay. Then write it accordingly. If there is any query, you can ask directly, or we can wait. Sorry? Then you can. So that's why it is important that the sequence is available for the gene you want to study. Paste it somewhere in Word, Notepad, or anywhere. So with this, you have done these three things. First of all, you have uh, you now know how to select an appropriate vector, and from where you can source it, and you can choose on the criteria which I have told earlier. And after that, selection of target gene, that depends on your work. what gene you are going to target, and the factors affecting the selection of target. I have mentioned that the genome sequence, gene sequence should be known, and it should have a PAM region as well, which we'll discuss in the later section. And here you also have this part, you have covered this part, retrieval of target gene from any databases. Similarly, there are specific databases as well, like if you don't get any sequence in NCBI or some these first-hand databases, then you can go for specific databases as well. Like I can give you example of plants because I work in plants. So there is a Soul Genomics, and where you can find genomes of Solanaceae family, and SPUD database where you can easily find about Solanum lycopersicum and Solanum tuberosum. So for Solanaceae, it's Soul Genomics, and for potato, it is SPUD. And for Arabidopsis, you must have known the tear. Tobacco, you can, Soul Genomics, you can, tomato, you can easily, yeah, Soul Genomics, SOL and Genomics. So we have covered these part, and so what's next? The next is designing our single guide RNA, and for which you should, like I have mentioned earlier, the sequence of the gene should be available or known to you. And second is the presence of PAM, protospacer adjacent motif. In our case, we are talking about Streptococcus pyogenes, so the PAM is NGG, and depending on the variant of Cas you are using and the species from which it has been derived, the PAM changes. Like earlier I have mentioned you a table with the different PAM. So our PAM region should be adjacent to the target region, and our target region should be in the CDS, which you have retrieved. And the third is that while designing the target area or target region, there should be no off-target effects, like your target shouldn't cut some other gene. It should be specific, like how we design primers, if you are aware of that, that it shouldn't be able to bind somewhere else. So it has to be specific, so no off-target effect should be there. And as I have mentioned earlier, that the target region should lie in the exonic region, so that's why we have taken the CDS part. And so earlier we can also design this target region manually. Now we have a lot of softwares or online tools available, but basically what they does is, in your target region you provide the sequence and they find this PAM. Like you can manually find this NGG, suppose we are talking about NGG, you can manually locate NGG in your target gene and in the CDS of the targeted gene, and adjacent to that you can select the 20 base pair target region which you want to target. And after that you can take the 20 base pair part and you can blast it with specific to your organism, and you can see that where else that target region is binding. So that's the basic background of these tools. These tools are doing that for you, so that you don't have to manually locate for these PAM, because sometimes the CDS can be long and it would be not efficient if you manually locate these PAM region yourself. So these softwares do them for you. So this is a list. So these are some of the popularly used tools which we use, like ChopChop, CC-Top is there, CRISPR is there, and the one with Synthego CRISPR design tool. So these are different tools, and they all have different key features which you can study later. Like some of them are only, you can submit your target gene with up to 500 base pair limitation, and some give more good graphical projection of your target genes. So we'll come to this later. So you can we will start with this chop chop. This is one of the tools. You can just type chop chop CRISPR in Google, or let me be with you. You can just type chop chop and CRISPR, then you will be able to find this. And you can click on the first link. Okay, all of you have reached here. So in this box, you can either paste the accession number or your target gene itself. So in our case, we will paste the target gene. So we will select, we will click on this here, paste target. So with this, after clicking on this, we can simply paste our FASTA entry here. So the sequence you have copied in Word or Notepad, you can just copy and paste it here. Okay, so in the next box you can see, you can select which organism you particularly want to target. So in our case, the sequence is of Arabidopsis. So we will search Arabidopsis athaliana here. Or you can just type that, smart Arabidopsis. Selected. Okay, so our demo sequence is of Arabidopsis thaliana, so we have selected Arabidopsis thaliana, and depending on your organism you can select the organism here. And after that we are focusing on that PHSC and PKSC 401 vector, which works on Cas9 variant. So we'll select CRISPR Cas9, and similarly there are different variants available, and different tools have different variants, like some of them even include the latest versions, latest endonucleases as well. Like this one even has TALEN, so we'll select CRISPR Cas9 here, and you can select the purpose, like you want to knock out, knock in, and I'm not sure that whether this will affect our target prediction, but we will just select knock out here. And after filling all these things, you can just click find target sites. So it will take some time. You can click and wait, but for your reference, I have already taken a screenshot of it. So like I have mentioned about these draw boxes where you can select the filters according to your need, and after completion you can get a result something like this, results of something like this. We can just wait. So that's why we are waiting. So it takes some time, so because you have taken different genes as well, so we can wait. So you will get the result something like this. This is the graphical representation. Here, this these small boxes represents the target sites. If you hover your, all of them are target sites. They are color coded according to their efficiency. So
All of you have gotten your results or not? Yes.
yes, I'll just come here once everybody is on the same page. So uh, एक ही result like, please specify. Okay. What target sequence you have selected, or and how long it is? That also. But how long it is? If the CDS is short, then it is possible. Okay.
एक की और सेम प्रॉब्लम है सर अगर सारे सेम ही कलर के आ रहे हैं तो नहीं उसका इम्प्लीमेंट करना
मैं आता हूँ इस पे भी. तो बेसिकली
एक बार इसके लिए मैं ट्राई करती हूँ तो पांचवा फॉर्मेट ही इससे नहीं नीचे स्पेसेस है
the sequence should be continuous, otherwise it will identify it as a small sequence. So like in this here in the graphical representation, uh, this is based on their location. Like below you can see the scale. Our CDS is which I have taken is approximately 1500 base pair. So that's why you can see this stretch here. This goes up to 1400 and further. So based on their location, they are there, not according to the list here. The list is according to their efficiency. Your FASTA sequences should be continuous without any disruptions or without any unrecognizable character. Because there are other parameters as well, like the mm0, mm1. So I'll just come here in a minute. So the list is not like that. The top target sequence you get, you can't just simply do that, because there are other factors as well, like these are mismatches numbers, like mismatch 0, mismatch 1, mismatch 2 and mismatch 3. These mm0, 1, 2, 3, and these represent like if with 0 mismatches it binds to a uh, uh, any other gene, like with this target sequence this binds somewhere else, like not where it's supposed to be, it binds to one other place with this. And with mismatch 1, like in the, like here you can see that this target with 3 mismatches bind in one position somewhere else, and these all results are interpreted differently in different softwares or different tools. So like in this list with this example, according to me the fourth one, with the rank 4 is the best one, because with mm0 it doesn't bind somewhere else, it's 0 in all 4 of these, mm1, 2, 3 and this. And it's also in the positive sense, like the strand is the positive strand, and you can see the genomic locations as well here, like it's in the 110107 position of the provided CDS. So similarly the efficiency is also not that bad, like for the first one it's 78, and for the fourth one it's 71%. So I think this will be a good target according to me. And earlier I have also mentioned that this is the scale, and depending on the length and the position of the target, they have been represented here in this section. Okay.
Sir, even his self-complementarity should be 0.
Yes, these are some also some parameters which you need to look for. Sorry? No, no, no. It's more of like when you design primers, you have to follow a particular range. It shouldn't be too high or too low, because that will hinder in further steps, which will be. This will go, because this is in the range between 40 to 60. It shouldn't be too low, but it either shouldn't be too high. So this will come into picture when we'll design primers. So we'll come to this. No, not necessarily. It can be negative strand as well, because our CRISPR RNA or this target region will simply bind at the particular location, and the Cas9 will introduce a nick there. So it doesn't matter whether it's in positive strand or in the negative strand. So you can choose either in the positive strand as well and as in the negative strand. So let me conclude everything. So yes. These are mismatch number, like with one mismatch your sequence is binding with some other target, like in other region. So I think this tool here is making it look a bit complicated, but when we will use a different tool like CC top, there it is more in a understandable way. But according to me, these all should be 0, then it's good. Like with the MM3, like I have shown you example like here, starting from there with three mismatches, our sequence bind with one off target. That's what it is showing. But with a different tool, I think you will understand this better. So these are all parameters depending on the tools, how they represent data and how they, some tools are easy to understand and I think it's also good. So if there is any question regarding this tool, I can, yes, please. Yes. Yes. Everything is red. You have taken CDS sequence. Okay. I don't think there should be any problem because it should be CDS and uh, here they represent intron and these things like here mentioned in the top left corner. But as we have taken the CDS itself in the first place, so the color coding sometime will not match according to this. But in some, like in this tool, we can use the normal genomic sequence as well, but in some tools there is a limitation that we have to use the CDS part. So that's why I have told you to take CDS consistently throughout. So the color coding, I don't think that this will be a problem. ये ये जो टॉप लेफ्ट में आपको वो दिखा रहा है बॉक्स कि एक्सोनिक प्लस एटीजी है उसमें रेड हमारा टारगेट के लिए वो बता रहा है परफेक्ट बट वी हैव टू वी डोंट completely rely on this. We have to look ourselves as well. Like I have told earlier that the rank one is not the one which I will take if I want to continue with this. So I will take the fourth one with this PIF3 example. So according to me, the fourth one is the best option. Yes. Yes. So, and one more thing, you can clearly see that our PAM was NGG. So you can see that with every target sequence at the last, we have NGG, like N can be anything. So here we have CGG, CGG, TGG. So, and upstream to that, there's our 20 base pair region. You can count, you don't have to count actually, this is 20 base pair long. So you can just click it. We are not representing it. The software is doing itself as we have taken it, as we have initially taken the CDS. Here we can also take the genomic sequence as well. So based on that, it can differentiate between the exons, introns, and that part. But as we have taken the CDS,
If you're asking about the green, yellow and red, तो वो
soft,
efficiency के basis पर
yes.
जो
So
part वो साइड में बॉक्स बना हुआ है ना उसको एक्सॉन लेफ्ट कॉर्नर लेफ्ट अपर लेफ्ट कॉर्नर में वो बना है ना लेजेंड बना है वहाँ से समझ आएगा.
okay. So So we don't usually rely on a single tool. We always cross check it with some other tool. So like here we can, we have selected this one, you can just highlight it or okay. So this was our result page and I have talked about the ranking according to the software. The first one should be the ideal target, but if we look on these aspects, like the MM0123 values, and according to that, the rank fourth one should be the target according to me, and it also has a great efficiency of 71%. So that's about this chop tool, and after that we'll come to this tool. This is CRISPR. You can just search CRISPR, CRISPOR, CRISPR. You have to select this one, UCSC one, I think. Yes. Yes. So here the interface is quite different. So, but the basic remains the same. There's a box, you can just paste your sequence here, and here's the drop down menu from where you can select your organism. And since like this sequence is of Arabidopsis, and here it's showing the version 9 of an old version of the genome assembly for Arabidopsis thaliana. So you have to take these things also in mind when you are designing the target site, because if we select this, the genome assembly is very old, like it was uploaded on December 2012. So with the updates, the genome assembly also changes, so we prefer to use a newer genome assembly. So you can screen out these tools. So that's why I have provided you with a list of tools. So that will vary according to you, your target organism and the available genomic version for that. So in this drop down, again we will just simply select 20 base pair NGG that's already selected by default, which is specific for Streptococcus pyogenes Cas9, and we'll just paste our sequence here. Okay, and in this step 1 box, here it is clearly mentioned that you shouldn't upload anything greater than 2300 base pairs. So different tools have their different limitations and advantages, disadvantages. And once you have tweaked all these and filled all these boxes, you can just submit it. And the result will vary accordingly, as this one is using an old assembly for Arabidopsis. So I'm not sure that we'll get the same result, but let's see. I can come down. The one with the latest version, like ITG4, I think is the latest one. I think so. I think so. In Soul Genomics, it's the latest one. But if you are using the NCBI one, then you can select the version 1.2, I think. Yes, that all depends which database you want to rely on. You have taken the same sequence. How are you managing to get different results? Okay. So everybody got the results, or we have to wait? Okay. Everybody got the results list. So we can ignore this part, which I have got, as we are just here for the demonstration purpose. So here it is clearly mentioned that our sequence is 1575 base pair long, and it have detected 155 possible target guide sequences or target sites, and here's they have clearly written that the green is the one with the more efficiency, with the more specificity, precisely. And in the table they have their own score, which they have given here. Their scoring matrices can be read in their manual as well. So if you want to read that, you can read. And here again we have mismatch 0, mismatch 1, mismatch 2, mismatch 3, and mismatch 4. And here with 4 mismatches, it's binding with 2 other off targets, like I was telling earlier, that this means that I think this is clear now. Like if there are 4 mismatches, then it can bind with 2 off targets which are not our target gene. So this means that. So as this genome in this tool, the genome assembly was old, so I'm not sure that our result will align with the previous tool. So, and besides this, it has given some additional information as well, like the information of the 2 off target sites is written here, like it is in chromosome 1 and chromosome 2. And this gene lies in chromosome 3. I know this part. So here we have 2 off target sites, one in chromosome 1, and it's also giving the location as well. So, and besides this, it's also providing the enzyme sites which are present in the target region, that could be helpful while designing the primer specific for that, or you can just simply skip it. Like when you will design the vector, when you will insert this target gene in the vector, you will need to, you don't want the same site, same restriction site for which you will be digesting the vector. So you can check that also, and this tool is also providing the enzyme sites present in the sequence. Like for example, if we are using BSA1 restriction sites for designing the construct, and if the same sites are available in this target region, that will be a problem. Yeah, suppose any other sites. Basically, I just made an example so that you can. So this was all about this tool, and one more tool I want to show you is CC top. You can just type CC top CRISPR and you can select the first link. Here also you have to paste your sequences, but here's the catch that you can upload only 500 bases here. So that's the limitation of this tool, but I think that the graphical representation is much better in this tool. So that's my personal thinking. So maybe for you some other tool will work. So you can just select the first 500 bases, and here you don't have to paste it in FASTA format as it is written in brackets here. Like I have just randomly taken a small part of the sequence, which is probably the exon 1, and after that I'll paste it here, and similarly there is a drop box and you can select what organism derived Cas you are using, as we are using the Streptococcus pyogenes as an example. So we will be selecting NGG here, and after selecting that you can also tweak that whether you want the target, what length target site you want, and we have set it to the default, that is 20. And here in this box you can directly search or find your okay. So you can see that the genome assemblies are different in all these tools, so you have to search for the tool which have the updated ones, so that will be relevant. So after selecting all this you can just submit, and it will also take some time. Okay, so please let me know when you get the results obtained. Okay, so here the color coding is very simple, or I use it quite often, so that's why I find it more easy. So the blue one, the targets here in this region, simply this is also the stretch, like we have approximately taken our sequence less than 500 bases. So this is the table, and this is showing the length of the stretch we have pasted here, and based on their position these boxes are made, and they are labeled as target. T12 stands for target 12 based on their efficacy, sorry. And after that the blue ones are the with the high efficacy score, and with gray one here, these are the ones with medium efficacy, and we don't have any red box here. If we get red box, that means that have a low efficacy score. So if you further scroll down, you can just find a table like for T1. You can just simply look here, and it has given a list of off targets, and it has also, like these here are off targets, like the target region is also binding to that site, and here they are also saying the mismatch score, like with mismatches 4 it is binding to this region, and this is the chromosome location on chromosome 4 from this number to this number. It is also providing that coordinates, and it's also telling that whether the off target is in the exonic region or the intergenic region or the intronic region. It is also color coded in red, yellow and green. So red is given to the exonic region. That means you don't have to take this, or you will, otherwise you will have off targets. So, and it has also provided gene IDs for the off targets, the gene which it has been targeting. So I find it very useful, so I use this tool for my work, but simply you can select based on various other parameters. So simply, so according to this, the T1 is not an ideal candidate here because it's binding with a lot of genes of targets precisely, and even in the exonic region. So we can just scroll down, and similarly with T2 we have 5 off targets which are binding with the mismatch of 4, and their score is also low, like the score is from 0 to 1, so 1 is the highest score and 0 is the lowest. So anything greater than 0.7 is higher score according to this tool. So the T4 we got has a higher score, but here it has also 4 off targets and it binds to the exonic region of those off targets. So we will not select that. I hope now it's clear that how different tools interpret data, and some are easy to read and some are a bit complex depending on how you find it. So if your target region that's predicted and it's binding somewhere else, and if it's binding in the intergenic region, then you can go ahead with that target as well, but that depends only in that case where you are not finding any other targets which doesn't have any off targets. Understand what I meant?
सर मतलब मिसमैच क्यों उधर जीरो होना चाहिए सर यहाँ पे मेरे टाइम 4 दिखा रहा है लेकिन उसका स्कोर हाई है
बेसिकली कि अगर आपको कोई भी ऐसा टारगेट नहीं मिल रहा है जो जिसके ऑफ टारगेट्स नहीं है, जैसे यहाँ पे बहुत सारे हमारे टारगेट जो इसने फाइंड किए हैं उन सबके ऑफ टारगेट्स हैं, ये टेबल वही शो कर रहा है। अगर बहुत सारे आपको ऑफ टारगेट्स मिल रहे हैं तो आप फिर फिल्टर आउट कर सकते हो कि जिन ऑफ टारगेट्स में इंटरजेनिक रीजन पे वो बाइंड कर रहा है उनको सिलेक्ट कर सकते हो। तो आपको, अगर ये तब का केस है अगर आपको कुछ भी नहीं मिल रहा है सपोज, otherwise you have to select only the target region which doesn't have any off targets. Okay, so if there is any question I can take that, or we can just come back to our slides. CC top? CC top? Okay. The table you are seeing, wait for a second. Here. This table shows the off targets. Like it shows 5 off targets with if we talk about the target 14, if you are using the PIF3 as an example. Clear? It's also providing the gene IDs as well from of the off targets. So with this we have successfully used these three tools, ChopChop, CRISPR and CCTop. All of them has their different user interfaces and their different representations. So any other questions or doubt till here? Yes, please. Okay. Yes.
और इसमें ये वाला ऑफ टारगेट नहीं है, सो सर यही वाला चूज करना है, मतलब
नहीं, आप इसको नहीं सिलेक्ट कर सकते क्योंकि ये जो जितने जीन्स हैं ये एक्सोनिक रीजन में बाइंड कर रहा है.
ओके,
हाँ जी, तो
तो
आप
हम इंटरजेनिक वाला लेंगे.
नहीं, हम ये लेंगे ही नहीं. टारगेट 42 हम कंसिडर ही नहीं करेंगे क्योंकि ये बहुत सारे ऑफ टारगेट्स दे रहा है और मोस्ट ऑफ द उनके ऑफ टारगेट्स एक्सोनिक रीजन में बाइंड कर रहे हैं. तो आप क्या करोगे इस केस में? नेक्स्ट स्ट्रेच लोगे. हमने सिर्फ 500 बेस पेयर लिए थे, तो आप नेक्स्ट 500 उठाओगे.
ओके,
We can also do that because in this tool there is a limitation that we can only upload 500 bases. So
सर टारगेट सीक्वेंस में ये ब्रैकेट क्यों है?
ये मिसमैच स्कोर है कि इस रीजन में हमारा मिसमैच होने की वजह से आ रहा है वो. मतलब we can't take it in simple words. We can't take this target 37 because it has two off targets and it binds to the exonic region.
मीन्स मैक्सिमम अगर होगा तो सब इंट्रोनिक या इंटरजेनिक में करना पड़ेगा.
हाँ जी. So with this, I think you have learned today how to select an appropriate vector for your study and from where you can get the vector and how you can retrieve and select a target gene. That depends on your study, and basically I have demonstrated how you can retrieve the CDS from NCBI and you can similarly retrieve it from any other database specific for your organism and work. Same retrieval of target gene and designing of single guide RNA. So using these tools we can design target site, and the next step will be taken care in the tutorial 7 in the coming I think today is tutorial 7, so that will be covered in the next tutorial. So thank you for your attention, and if you have any questions you can just ask, or you can maybe connect later if you want to apply this CRISPR-Cas system to your work. You can just email or connect with Dr. Kashmir Singh. We are happy to help. So any other questions? Okay.